Hello everyone, and welcome to Occupational Therapy 245, Introduction to Research Methodology. Today's lecture is on quantitative study designs. And we will actually be having two lectures on quantitative study designs. This one is larger scale studies, and then there will be a separate lecture for smaller scale studies. So stay tuned for that. My name is Melissa Kay, and I'll be your instructor. Let's go ahead and get started. Our objectives today are to identify the different types of quantitative designs. We're then going to look at describing them and seeing what characteristics they have, explain the advantages and disadvantages of a variety of designs, and define key terms for quantitative study designs. So we've looked at this pyramid before, and we're going to revisit with the goal of actually looking at some of the different types of studies in more depth. And please refer back to this as you go because it gives you a lot of information about where studies fall with regard to rigor and the quality of evidence. There's two places that we're going to be referring to with regard to evidence and quality of evidence. The first is the Center for Evidence-Based Medicine at the University of Oxford, and the other is the AOTA, Evidence-Based Practice and Research for the U.S. So there are there's some links for you in the presenter notes. Please check those out for more information. All right, and let me just adjust this slightly for you. This is a visual model from Grimes and Schultz, and it looks at how we determine, how a study design works, basically. And I'll need to move it around. Give me just a sec to make it a size where you can actually see it a little bit better. Is that going to work? Not, yeah, maybe. Let's see. Sorry about that. It's just getting cut off on my screen. Okay, there we go. All right. So it's an algorithm to identify different study designs. And you see that at the top it asks if the researcher actually allocated the subjects or participants, we call them both, to exposures, and that means to an intervention. If yes, it's an experimental study. Was there random allocation to one of two groups or more groups? If yes, a randomized control trial, and if no, a control trial. Now, on the right side of the screen, if the participants were not exposed to an intervention, then it's an observational study. Were they compared? If yes, it could be analytical or temporal directional. And if no, it's a descriptive study. And then you can see at the bottom that there's 3 different types listed. So one thing that I want you to get a little bit used to, and I know that for students, and myself included, of research, it can be a little bit frustrating, is that studies in different places are called slightly different things, and so we need to be a little bit flexible about our understanding of which study is which. And if you have some confusion, please bring those questions to class, and we'll be going over it in class as well, okay? But do keep an open mind that studies may be called slightly different things. There may be slightly different opinions about which level of that pyramid a study goes at, mostly like one level up or down, and that's just to be expected with research. So frustrating, but true. All right, so let's move on. We're going to talk a little bit about research design terms, and then we will go into the different types of studies. So there's something called exposure, which is also the independent variable, or IV. And this is the variable that can be controlled, selected, and manipulated. In other words, it's the factor that can influence the outcome, and therefore it is the intervention or the thing that gets changed to see how participants react to it. The outcome is also called the dependent variable, or DV, and this is the response or the effect from the treatment or intervention. So the outcome depends, right, dependent variable, on the independent variable or the treatment. The next term is actually has definition in its title, but it is operational definition. And what this means is that within a research study, there may be terms that the reader is not familiar with, or that you as the researcher define in a very specific way for the sake of your particular research project. So an operational definition is a description of a procedure, action, or process by which a variable could be observed and measured. So it's really getting the researcher to hone in on what exactly they're talking about. Many research studies and your literature review will have a number of operational definitions, and all that means is that you're giving a very specific description or definition to the terms that you're using in your study. Randomization, which we talked about a little earlier in a previous lecture, is the process that participants in research studies are assigned by chance, right? So random, to separate groups, and then those groups are given different treatments or interventions. So it can be achieved through a random number generator, a roll of a die, pulling names out of a hat, etc. But basically what it does is it gives every member of the sample an equal opportunity to be in either group in the study. And by doing that, we reduce the error. In other words, we reduce the likelihood that the results that we see are due to our study design as opposed to being due to the intervention. All right, our next term is confounding variables. And confounding variables are anything that can act on a participant in a study that is not the independent variable, right? So it's not the treatment, it's anything else, and it changes the outcome of the study. So they're variables that confuse the outcome of the study. So in this example, our independent variable is alcohol consumption. Now, we're not feeding people alcohol, but we're looking at a sample of people who drink alcohol, and we're saying, what is the relationship between alcohol consumption and lung cancer, right? Now, the thing that confounds this is that when people drink alcohol, they may be more likely to smoke, or they may be smokers to start with, and so alcohol consumption and lung cancer may be related, but it may be related because of the connection with smoking rather than the connection with drinking. Our next term is bias, and bias is any tendency which prevents unprejudiced consideration of a question. So it's anything that will skew or cause the results to deviate away from an objective truth. In research, bias occurs when systematic error is introduced into sampling or testing by selecting or encouraging one outcome or answer over others. So what does this mean? So in research, if we undersample the population, we may not have enough people to actually get an honest outcome that represents the truth of the sample. And therefore, we wind up thinking that we have an answer, but the answer is not actually very accurate because we in reality, we needed to sample a lot more people. And there's a number of different types of bias. There's selection bias, which has to do with sampling, and then measurement bias, which has to do with measurement. In your presenter notes, there's a very good article from the from the NIH, the National Institutes of Health, about bias. And we will be looking at that more as we go. When we read research articles, it's very common for authors to actually acknowledge any bias that may have crept into their study rather than just skimming over it. All right, our next term is blinding. And what this means is that we conceal the status of the participant from either the participant themselves. So we might provide, if we were in medical research, we might provide a placebo or a new drug, right? And the participant doesn't know which one they get. From the researcher, so sometimes we have some researchers actually do the study, and other researchers analyze the results, and they don't know which participants were in which group and who received what sort of intervention, and that keeps them honest and objective. Or we might have blinding from both. A statistician or study team could also be blinded. So it's a way to remove any tendency, unconscious or conscious or whatever, that skews the research in any way. And it's just a measure to increase rigor. All right, so now we're going to move into experimental designs. As was noted earlier, the randomized controlled trial, also known as the gold standard, and at the top of that pyramid is the only design that can draw causal relationships. So in other words, it is the only research design that can say this independent variable caused this outcome. The characteristics of RCT are randomization, which we have talked about, right? So all members of the sample have an equal opportunity to be assigned to any of the research groups, right? Research study groups. We also have manipulation that basically means that there's an intervention that the participants are exposed to. And we have a control group. So the control group is used for comparison. In occupational therapy and other healthcare fields, it is often unethical to have a control group that does not receive any intervention at all, right? So say we have a group of cancer patients and we're trying out a new immunotherapy, right? It would be super unethical to give it to half the people and for the other half the people with cancer say, well, you're not getting anything, right? So often what happens in healthcare studies is that we have the current standard of care and we use that as the control. So it may be in the cancer example that the experimental group gets the immunotherapy and the control group gets chemotherapy or radiation or a combination. So whatever is standard for the treatment. So that's an RCT. All right. So back to our visual model. I don't know why I'm having so much trouble talking today, you guys, but I really want to get this recorded and out to you. So we are on the left-hand side. We have an experimental study as noted by that red arrow going down, and it's a randomized controlled trial. All right, so we're going to be talking a lot about something called the well elderly study. And this was done by Florence Clark and colleagues at the University of Southern California. And what they did was they looked at the effects of occupational therapy intervention on a sample of older individuals. So this is a visual model of that experiment. On our left in the circles, we have the study sample, and then we have the population. Remember, and there's and the population is greater than the sample. We have random assignment, and then the experimental group got OT intervention. There was a social group as a control, so they were already providing social interaction as part of living in this one particular place. And there was no treatment. So in this case, the no treatment control happened because there was people that weren't participating in the social groups. They were just living at this place, right? They measured the effects of occupational therapy, social groups, and no treatment control at 1 month and 9 months. So a pretty long study. The social group, the OT group engaged in 2 hours a week of group OT, 38 sessions, and 9 hours of individual OT. The social group had 2.25 hours per week of activities to encourage social interaction. And here is, I know it's probably a little bit hard to read. You can read it on your screen for sure. But I described the study. Here's the synopsis of it. And the results were that the OT treatment was found to be efficacious for the quality of interaction for older adults. And as you see under the results section, they measured a whole bunch of different aspects of function in older adults. The study concluded that there are significant benefits to OT, and it was found across various health, function, and quality of life domains. The control groups tended to decline over the study interval, and so the results suggest that preventative health programs based on OT may actually mitigate against the risks of older adulthood. So that's a pretty strong finding. You can certainly look this study up. It was done in 1997, and I've provided a link for you. Now let's turn to a non-randomized controlled trial. So this is also known as quasi-experimental design. And in this, as compared with the RCT, there may be a lack of random assignment and or a lack of comparison group. And it often involves non-equivalent groups, so groups that are not matched necessarily on all aspects of their inclusion in the group. So they may be a wider range of ages. They may have more than one condition, right? They may have some kind of difference that makes it impossible to test for a causal relationship. Now, remember in the RCT, that was the only design where a causal relationship could be determined. In the non-randomized controlled trial, a causal relationship cannot be determined. And again, we take our visual model, and you can see that it's in the experimental section and the controlled trial, but not a randomized controlled trial. All right, so there's a number of other types of experimental study designs. The first one is between group comparison, and typically what this means is that there's a pretest and a posttest and a control group. So the pretest posttest design is an experiment where measures are taken before and after treatment. The design part means that you're able to see the effects of some type of treatment on a group. Pretest posttest designs may be quasi-experimental, which means that participants are not assigned randomly. There are also repeated measure designs. And this is a research design that involves multiple measures of the same variable taken on the same or matched subjects, either under different conditions or over 2 or more time periods. So we may do repeated measures in a longitudinal study in which change over time is assessed. So we may take a measure at 1 month, 6 months, a year, 3 years, 5 years. So that is repeated measures. And then crossover, sorry, I've flip-flopped my explanations, but we're in within group comparison, right? So between group means that we have to have two groups. Within group means that we look at the same group at different points in time. And a crossover design is a repeated measures design so that each participant receives different treatments during different time periods. So for example, patients cross over from one treatment to the other during the course of the trial. And for some of you, you participated in my homework study last semester, your first semester in the program. And there were those of you that were in the concept map homework group, and those of you that were in the question and answer homework group. And halfway, you switched over. So it's a way to get more information, but still using one group of participants. This is a visual of the pretest, posttest control group design. And notice that there's the study sample. Homogeneous groups means that they are like on the measures that are important. There's random assignment to an experimental group or a control group. Then there's a pretest, so we measure some aspect of behavior or skill and get a baseline. Then we do intervention, and the control group either receives a placebo, comparable intervention, or no intervention at all, again, depending on the ethics of the situation. And then we have a post-test, which gives us our outcome. Going back to the well elderly study, we had the study sample. We actually had three groups, right? So remember, we had the OT group, the social group, and no intervention. And we had random assignment, making it experimental. We had a pretest, and then we had a post-test. And I believe that was at 1 month and 9 months. All right, so within group design, and we've been talking about this a little bit, we have one group of participants, and each participant acts as their own control. So the effects are observed within a participant as opposed to between participants. Treatment effects are associated with the differences observed within a subject across treatment conditions rather than between subjects across randomized groups. So sometimes it's easier or more feasible to actually have one group of participants and we try different stuff with them, right? So it's ambitious and it can be difficult to find enough participants to do a two-group design, and then even more difficult to engage in random assignment. Sometimes it's because of where the clients are, what kind of treatment they're receiving, etc. So a number of reasons why it may not be feasible to have a two-group design. Now, of course, having one group of participants as opposed to two that are randomly assigned to groups is not as rigorous or as strong as having two groups. So here are some advantages and disadvantages about within group design. And I think I've gone over a few of them. So advantages, you have increased control because there's your one group. You have an equivalent comparison. So each person is experiencing an intervention or two interventions, and we're comparing them, right? But the person is still the main unit. So you can actually compare intervention A and intervention B quite easily. And those are the two main advantages. The disadvantage is possible practice effects. So if we have somebody do intervention A, then intervention B, and intervention A and B are kind of similar, then they might get better just because they had the opportunity to practice with intervention A, right? Rather than measuring only the results of the intervention itself. There's also a learning effect. So over time, they may learn what's happening in the study or with the interventions, very similar to practice effects. And then there's carryover effects. So how long do we wait between providing intervention A and intervention B? If we don't wait long enough, intervention A, the residual effects of it may still be happening. And so when we implement intervention B, we're actually getting kind of the leftovers or the residual of intervention A. Okay, next up, one-way repeated measures design. So we have a study sample, we have 3 interventions, and we have 3 points of measurement. And this was actually a study by Newman in 1998. It was hip abductor muscle activity as subjects with hip prostheses walk with different methods using a cane. So the interventions were different methods of walking, and they measured the subjects or participants after each intervention. In the well elderly study, there was a semi-crossover design, and it consisted of Group A intervention, Group B control, and this was a portion of the study. It was a huge study. So this was another portion of the study. Group A got an intervention and was followed. Group B got an intervention and then was followed, and they were tested every 6 months to see how they fared over time. So it contributes to the rigor of a study if you have a long time, in this case 2 years, and you can follow people and potentially do a crossover. So Group A gets the intervention first, then Group B gets the intervention. And Group B in month 1 is a control group, but then you can see in month 6 that Group B gets the intervention. So we stagger the intervention amongst the groups. All right, so it's that time for you to take a break, get up, stretch, have a drink of water, step away from your keyboard and your mouse and your computer, and come back in a few minutes, and I will see you real soon. All right, everyone, welcome back. We're going to go on with the second half of this lecture, and we're going to be looking at experimental considerations. So we're returning to some information that we discussed a little bit earlier, the learning or practice effect and the carryover effect. So once again, and these are called order effects, and it's important that you know about them. So the learning or practice effect, again, is when the participant repeats the task over and over, their performance improves. The carryover effect, again, is when the subject or participant is exposed to multiple conditions, the effect of being exposed to the intervention or condition is reduced because there is not enough time between the different treatment conditions. Within healthcare, you would think, okay, well, let's just do a whole bunch of randomized controls trials, right? It's the best. So there's advantages and disadvantages to experimental designs. And this is like taking what the ultimate is and applying it to real life, right? So cost is definitely a consideration. Costs money to run research, especially high quality research. The participant requirements mean that you have to cast a really wide net, and it can be more difficult to recruit. Also, and this one is particularly important for OTs, heterogeneity of clinical populations. And what that means is that, for example, I'm in pediatric practice, right? So I could do a study on kids with autism. But what we know about kids with autism is that they all look different, and they all have different constellations of signs and symptoms and behaviors. And so even if we're measuring an intervention, right, a particular intervention, not having participants who are very much the same on all other measures, right, and are very, very similar to each other can skew a study. So finding a homogeneous sample within a clinical population is extremely difficult. There's also ethics. Again, the control group, no treatment is one aspect of ethics, but other aspects of ethics can crop up too, and we'll talk about that when we go through our ethics unit. Another disadvantage is that generalization to individuals may be difficult. So we have this high-level study with a lot of people, and then we have our client. And generalizing from this study to our client still may be very difficult because our client isn't the same as the rest of the sample, right? So that can be a drawback, kind of a mixed bag drawback. Also, I've said before that when we have a large sample, we find statistical significance, which is what we're looking for. That is different than clinical significance, right? So what we're after is clinical significance, but with an experimental design, what we may get is just statistical significance. There's also potentially some compatibility difficulties with clinical practice. So having an experimental design means you have a lot of controls in place, right, to prevent confounding variables. But if you're doing a study in a clinical setting, what we want to be focused on is the health and welfare of our clients, not how much control can we have over the environment, for example. So the ability for clinicians or practitioners to do research is limited when we're in clinical practice, and that can be a challenge with experimental design. All right, so now we're on to non-experimental designs. And remember, these are the ones that don't involve an intervention or an IV. It's also called a descriptive study. And as I just said, there's no assigned exposure or intervention. And here we're on, again, with the red arrow on the right side of this chart. When we think about surveys, descriptive studies, we're often thinking about surveys. We want to describe characteristics about a population or about a phenomenon. It doesn't answer questions related to how, when, or why a phenomenon occurred, and we cannot draw a causal relationship. So I'm going to kind of hammer on this. The only place that we can draw a causal relationship is, I hear you saying it at home, the randomized controlled trial, right? Anything else, we cannot draw a causal relationship. So observational study designs can take many different forms. There's cross-sectional designs, case control, case crossover, retrospective cohort, and prospective cohort. And I've given you definitions and examples of all of these in your presenter notes. So please review them, and we'll talk about them in class. All right, and here we are back on our chart with a descriptive study with no comparison group and a descriptive study with a comparison group. And in that study, we would look at timing. So we're going to look at a few different kinds of observational studies. Cohort studies, there's an exposure and an outcome, and the study begins with an exposure. We follow the group for a set period of time, or two groups are followed over the same period, and these typically take years. So it's not that we have actually used an intervention, but for example, it could be the onset of COPD, right? Or the onset of heart disease. And over time, those diseases will develop and change, and we will measure over time what happens to the individuals. There's two different kinds of cohort studies. There's prospective and retrospective. So Retrospective is looking in the past, right? So we have the start of data collection, and we look back into the person's chart to see, okay, when was the onset, what's happened, and then we get to the present, right? We move from the past through a chart review up to the present and look at their outcomes. The other type is a prospective cohort study. And in this one, we start data collection now and we follow those people into the future to see what their outcomes are. So both are types of cohort studies. Throughout this section, there will be these slides that are for your reference that will simply have different summaries of different articles. And so you can have a look at it to see how are different types of studies characterized. All right, and this is just another way of looking at a prospective cohort, also known as a concurrent cohort. So in 2018, half the people are smoking, the other half are standing around next to them, and we're wondering what happens to them. And then in 2028, in 10 years, what happens to the people who are smoking? Notice there's these little horrible lungs on some of them. And then the people who are not smoking but are standing next to them, one person has developed lung problems. This is a retrospective study, and you can have a look at that. Now let's look at case control studies. So we have an exposure and an outcome. The cases and controls come from the same population, and we match the controls to the cases. So we select cases that are representative of all cases, and we select the controls so that they're similar to the cases on the potentially confounding characteristics. So if we think that gender might be implicated, and we have somebody who has muscular, who has MS, multiple sclerosis, we would then match the person with MS to the gender of the person who's in the control, right? And it could be gender, it could be age, it could be other conditions, it could be socioeconomic status, it could be the region in the US that you live, it could be any number of things, but the controls are similar to the cases on the potentially confounding characteristics. That's what's the important part of this. Here's an example of that, and it's using the MMR vaccination and its relationship to PDD. Here's another one, cross-sectional study. And now we're going to look at cross-sectional versus longitudinal studies. So a cross-sectional study, we have a group of people, we have subjects that are 20 to 30, 30 to 40, and 40 to 50, right? And we measure them at one point in time. In a longitudinal study, we have our group of subjects or participants. We measure them at time 1, time 2, time 3, right? So cross-sectional is one point in time. Longitudinal is over a long period of time. And in cross-sectional studies, we could have a new sample of people each time we do the study, but in a longitudinal study, we have to have the same people over time. Makes sense, right? All right, we're getting to the end of this, and here is a little bit of levity. This is a cartoon about a double-blind study. Slightly lame, but slightly funny. All right, so next week, which is next lecture, we're going to go over case series, case reports, and single subject studies. So the smaller types of quantitative studies. All right, that's what I got. Thank you so much for your attention, and please jot down your questions. I will be going over the high points of this lecture when we see each other, and I will see you in class. Thanks, everyone. Take care. Bye now.